my fellow viewers. Welcome to the Mr.Wanyoike, the digital learning platform. In front of you is Mr. Wanyoike himself, presenting a biology lesson. I am a biology teacher, and I'm going to continue with genetics as from the previous lesson, that is a topic in Form 4. And on the previous lesson, I introduced the topic, and in today's, I'm going to elaborate more of the structure of the chromosome, or the number of chromosomes which are found from one organism to the other. You must know that different organisms have different number of chromosomes, and as far as the number of chromosomes is concerned, we will be looking on the both somatic cells, the number of chromosomes which are found in their body or somatic cells, and also the number of chromosomes which are found in the gametic cells. To start with, what is a somatic cell? Somatic refers to the body cells. Or what about gametic cells? This refers to the sex cells, and the sex cells are the ones, for example, the sperm, and also we have the ovum, as far as in female is concerned. Then I'm going to look on the various organisms, and I'm going to give the exact number of chromosomes which are found in the somatic cells, and also the number of of chromosomes which are found in the gametic cells of different organisms. To start with, I'm going to start with the organism that is a cow. And as far as a cow is concerned, the number of somatic cells, they are supposed to be 60 as far as somatic cells is concerned, and the gametic cells, they normally have 30 chromosomes, and therefore the number as far as a cow is concerned, the number of chromosomes are supposed to be 60. That is, they are diploid. Diploid number is supposed to be 60 chromosomes in the cell and somatic cell, and in the gametic cells they are supposed to be 30. And as far as the second organism is concerned, and that is sheep, and when we come to sheep, the number of chromosomes which are found in the somatic cell or body cell is supposed to be 56. And therefore it is supposed to be 56 in sheep. And the gametic cell, that is the gametic cell, normally have 28 chromosomes, and that is haploid, meaning that a half of 56, because somatic cells are diploid, and while the gametic cells are supposed to be haploid. And as far as fruit fly is concerned, a fruit fly is an organism which was used by the father of inheritance, that is Gregory Mendel, and therefore this father of inheritance, during his study, his work, he branded various fruit fly in the lab, and that's why he was very successful in the work of genetics, or to explain more about inheritance and variation. And as far as a fruit fly is concerned, the number of chromosomes which are found in the somatic cell are supposed to be 8, and therefore the diploid number is supposed to be 8, and therefore the gametic number of chromosomes, the chromosomes which are supposed to be found in the gametic cells, is supposed to have the number of 8, that is exactly 4. And as far as maize is concerned, the maize plant is concerned, the number of chromosomes which are found in the somatic cell is supposed to be 20. Chromosomes are found in the somatic cell, while 10 number of chromosomes, or 10 chromosomes, are supposed to be found in the gametic cell. And what about Homo sapien? That is the scientific name for man. What about man? Homo sapien. And as far as the number of chromosomes which are found in the somatic cell is concerned, we have 46 chromosomes in the somatic cell. What about in the gametic cells? As far as man is concerned, is have the number of 46, and half of 46 is supposed to be 23, and therefore in the somatic cells there are 46 chromosomes, while in the gametic cells man have 23 chromosomes, and that is as far as the different organisms are concerned, we can make a conclusion that different organisms have different number of chromosomes in their somatic cells, and also have a different number of chromosomes in their gametic cell. Having said that, I'm going to look on another area, and I'm going to look on the structure of the DNA, or the gene. And as far as the gene is concerned, I explained it the previous lesson, and I said a gene refers to the unit of inheritance. That is the definition of genes. Genes refers to as hereditary factors. They are hereditary factors. Refers to the hereditary factors. They are hereditary factors, or you can say genes are units. They are units of inheritance. Therefore, that is the definition of the gene. And there is something else that we refer to as gene locus. And as far as gene locus is concerned, it is the position on the chromosome, which is occupied by genes. Or you can say the locations on the because it is a singular locus, location on the chromosome on the chromosome in which gene occupy, and that is what we refer to as a gene locus. According to a scientist by the name Oswald Avery, Avery, in 1944, he came up with the idea. He established that genes exist in form of nucleic molecule called DNA. Therefore, according to Oswald Avery, in 1944, he established that a gene exists in form of nucleic acid molecule called DNA. And those DNA is the acronym of DNA. That is the acronym of deoxyribonucleic acid. That is the acronym for deoxyribonucleic acid, and that is D stands for deoxyribo. There is O there. Ribo. N stand for nucleic and A stand for acid, and therefore DNA. And he found that DNA. He found that the gene exists in molecules that we refer to as DNA. And according to other two biologists by the name Francis Crick, according to other two biologists, that is Francis Crick and James Watson, in 1953. They established that, or they noted that the DNA structure is made up of 5 carbon sugar. Is made up of 3 components. Actually, the first one is 5 carbon sugar. The second one, phosphate molecule. That is, they found that in 1953, they found that DNA is made up of three structures. Number one, 5 carbon sugar, or pentose sugar. The second one, they found one phosphate molecule, and the third component was nitrogenous base. Nitrogenous base. They established that the gene, the DNA, that is the basic structure of the gene, that is the DNA, is made up of 3 components, that is 5 carbon sugar or pentose sugar. Number 2, the second component is phosphate molecule, and the third molecule is nitrogenous base. That is according to 2 biologists by the name Francis Crick and also James Watson. That is what they found out, that the gene is made up of those structures, those 3 components, that is in the year 1953. And also it is very good to note that there are 4 types. There are 4 types of nitrogenous base. It is very good to note that there are 4 types of nitrogenous base. 4 types of nitrogenous base, and the first type of nitrogenous base is called adenine. So types of nitrogenous base, we have the first type is adenine, that is denoted by capital A. Then we have the second one, we have guanine, denoted by letter G, and the third one is cytosine. It is denoted by letter C. That is, as far as DNA molecule is concerned, there are 4, and therefore the fourth one should be diamine, and it is denoted by letter T. Moreover, it is worth noting that the 3 structures, the 3 components, linked together to form a structure called nucleotide. And therefore, the 5 carbon sugar, I said the 5 carbon sugar, and the phosphate molecule, and the other one is nitrogenous base, they normally join to form a nucleotide, a structure called nucleotide. And therefore, the simplest structure formed whenever the 5 carbon sugar link with the 1 phosphate molecule and 1 of the nitrogenous base, it will form a structure called nucleotide. And therefore, several nucleotides, that is nucleotides, link together to form a DNA strand. And therefore, a DNA strand is the structure that is formed as a result of several nucleotides linking together. And therefore, after the DNA strand is formed, 2 DNA strand link, or we can say join or zip. They normally join or zip together, become zipped together, to form a molecule called double helix. And therefore, the structure which is normally formed as a result of two DNA strands linking together or zipping up together is what we refer to as a double helix. And that is how DNA structure exists, and therefore it exists in form of double helix molecule. And therefore the double helix molecule is the form in which the DNA structure finally exists. And as far as the linking is concerned, I can draw to show a DNA double helix molecule, that is a double helix molecule, and explain to you how it is formed. You can see that here I can draw one of them here. You can see that is one strand. Just like a zip, one part of the zip. Then the other one will come like this, like that, and you can see it is like a zip. They are zipping up together, and on one side I will have the nitrogenous bases. Nitrogenous base, they are there, and also on the other side I will have the, for example, I can have diamine, adenine, guanine, cytosine, adenine, diamine, cytosine, guanine, adenine, diamine, cytosine. And therefore on the other side you will have also the nitrogenous base, and as far as the other side is concerned, I will have diamine. You must note very clearly here that diamine, which is a nitrogenous base, will always link with adenine. Diamine, which is a nitrogenous base, will always link with diamine links with adenine. On the other side, the guanine, on the other side, guanine, which is also a nitrogenous base, will always link with the cytosine, and therefore diamine link with adenine, while cytosine link with guanine. And therefore, when we come to fill the remaining space here, as far as this one is concerned, you will find that diamine joins with adenine, adenine, diamine, guanine, cytosine, cytosine, guanine. Therefore, capital G there. Then we have adenine, diamine, diamine, adenine, cytosine, guanine. Then guanine, cytosine, adenine, diamine, and diamine we have adenine, and cytosine will have a guanine. And therefore, that is how double helix will appear. That is how a double helix will appear. And therefore, what about the process of zipping up? I can illustrate it using a different one here, using a different diagram here. You can see that there is also the second diagram here, and this shows how the double helix link together. For instance, we can have the 5 carbon sugar. You can see that how many sides does this structure have? 1, 2, 3, 4, 5, and therefore this is a carbon sugar, and this is a phosphate molecule, and at the end we have a nitrogenous base, and therefore this is what now we refer to as if I can cut this. If I can cut this up to that, I can have this being the nucleotide. This is a nucleotide, and therefore this one nucleotide, and this is another nucleotide. So this is nucleotide 1 and nucleotide 2. And this is another nucleotide. If I can break them like that, I can have several nucleotides like that. That is one nucleotide, the second nucleotide, the third nucleotide, the fourth nucleotide, the fifth nucleotide, and so on. And therefore, after linking this nitrogenous base, this can be diamine, adenine, cytosine, guanine. And on the other side, I said, and you have to be very keen here, that diamine always combine with adenine, and adenine combined with diamine, cytosine combines with guanine, and guanine will always combine with linking with guanine will always link with cytosine, and therefore if I can link all of them, if I can link all of them, I can have now what we refer to as a double helix. This is what we refer to as a double helix molecule, and therefore you can see that it is a long chain. It is a long chain of DNA structure, and this long chain of DNA structure is what we refer to as a double helix, because we have one strand on one side, one DNA strand on one side, and another DNA strand on the other side, and therefore the two strands will twist together or will zip up together to form a double helix molecule. Molecule. Having said that, I'm going to look on the roles of the DNA, the roles of the DNA. And as far as the roles of DNA molecule is concerned, as far as the role of DNA molecule is concerned, the first role of the DNA molecule, it is to store the genetic information. So the first role of the DNA molecule is to store the genetic information, and the second role of the second role of DNA is to enable the transfer of genetic information unchanged to daughter cells through replication. And therefore the second importance, and I think it is very, very important, I write them so that you can easily remember whenever you are asked in an exam. So the roles of DNA, roles of DNA. So the first one is to store the genetic information, to store the genetic information in a coded form. The second one is to enables transfer of genetic information unchanged to daughter cells through replication, through the process of replication. And the third one, it is to translate the genetic information into characteristics of organism through protein synthesis. There is N, through protein synthesis. Therefore, those are the three laws of DNA, and therefore, whenever you are asked in an exam, you write the roles of DNA. The roles of the DNA. You're supposed to write the first one being to store the genetic information in a coded form. Then number two enables the transfer of genetic information unchanged to daughter cells through replication. And number three, you are supposed to translate the genetic information into characteristic of organism through protein synthesis. And therefore, those are the three roles of DNA in as far as in living organism is concerned. And having said that, I'm going to the process of DNA replication. And what is DNA replication? What do we mean by the term DNA replication? DNA replication is the process through which DNA molecule forms an exact replica That is what we refer to as DNA replication, the process through which the DNA molecule forms an exact replica. That is what we refer to as DNA replication. And I'm going to demonstrate on how DNA replication comes about. And as you can remember, there is another diagram which I had drawn, and it was looking like this. And therefore, during DNA replication, what normally happens is that the double helix, that is the two pairs of DNA strand, unzip. And therefore, you can see them unzipping. You can see there is a very big gap. It is like they had zipped together, and therefore they unzip. And after unzipping, there is new strand develop in between. Two new strands develop at the center, like that. These are the new one. And as you can see that I said, as far as adenine is concerned, it will always link with diamine. So in the new DNA strand, there will be a diamine, and in the and this process is what we refer to as replication. And adenine goes, diamine goes with adenine, guanine, cytosine, guanine. That is capital G. Then adenine, diamine. Then we have adenine going with diamine. On the other side, we have diamine goes with adenine. And for this is the original template, the original DNA, the original DNA template. That is parent, the parent original DNA template, and this is the new. This one the new DNA strands form. New DNA strand. Those are the new DNA strand. And therefore, what I've said that there is that process of unzipping, and two strands develop at the center, and the genetic information from the parent DNA template is copied onto the new DNA strand to form the new DNA strand. And therefore, you will have adenine combining with diamine, cytosine combining with guanine, and guanine combining with cytosine, diamine combining with adenine, and diamine combining with adenine. And therefore, we have lead to the formation. We have lead to the formation of a new DNA, two new DNA strands. And therefore, what you are supposed to know is that this or parent DNA template, that is the original parent. You can write the original on top, original, or parent DNA template. And therefore, after that process have taken place, this parent DNA template will coil together with this one. That is the new one of the DNA strand. They will coil together, and they will end up forming an exact replica of the original DNA. And therefore, this likewise, the other DNA strand, this one, will coil or zip up with this one. This is the parent DNA template. They will also link or zip, or they coil together to form the new double helix molecule. And therefore, as the process will come to an end, having two identical DNA structures, and that is what we refer to as DNA replication. And therefore, we have come to the end of that definition as far as the DNA replication is concerned. Now, I'm going to look on the other aspect of DNA, that is the role of DNA in protein synthesis. Have you ever asked yourself, or do you know where the DNA structure is found? Actually, if you don't know, the DNA are normally confined in the nucleus of a cell. they are supposed to be found inside the nucleus of a cell. And therefore, you normally find that protein is not synthesized in the nucleus, but protein is synthesized in the ribosome. And ribosome, these are cell organelles. A cell organelles is a living structure that are found within the cell. And therefore, the ribosomes are found in the cytoplasm of the cell. And therefore, ribosomes are outside the nucleus. And therefore, the DNA requires a mechanism of relaying the information of the type of protein which it needs to be synthesized. And therefore, there is another nucleic acid which is involved during the process of protein synthesis. And therefore, as far as the DNA replication process is concerned, and therefore we are going to come up with another type of nucleic acid formed so that it can facilitate the relaying of the information from the nucleus to the cytoplasm, especially where the ribosome is found, so that the process of protein synthesis can start. And the second type of nucleic acid is called RNA. So those are the acronyms of ribonucleic acid. And therefore, the second type of nucleic acid is what we refer to as RNA. And as far as RNA, I've said they are acronyms for ribonucleic acid. Ribonucleic acid, RNA. That is also another nucleic acid which is involved during the process of protein synthesis. And therefore, as I have already said that DNA is confined in the nucleus, but protein synthesis normally takes place in the cytoplasm, especially at the ribosome. Those are the cell organelles which are involved in the process of synthesis of protein, or protein synthesis. And therefore, the RNA must be made. So the DNA normally form or make RNA so that it can take the information in coded form. Remember I said the information in coded form from the nucleus where the DNA is located to the ribosome where the activity of protein synthesis is taking place. And therefore, how do the ribosome, how are ribosome formed? I'm going to take you through the same process. I can use this diagram to explain how RNA is formed. And RNA is formed as I'm going to explain. Originally, we have a double helix unzipping. So the double helix unzip. So that is the first thing that you're supposed to know, that the double helix, the DNA, the two DNA strands called double helix will unzip. And after unzipping, it will develop a new strand. A new strand is developed in between. And in the new strand, the position of diamine is replaced by another nitrogenous base called uracil. Therefore, in RNA, position of diamine, that is T, is replaced by uracil. That is U, that is denoted by letter U. And therefore, you are supposed to know that the position of diamine as far as RNA is concerned is replaced by a nitrogenous base by the name uracil. And therefore, in this example, you can see there is adenine. Initially, adenine was linked with diamine, but in the resulting RNA, you will have uracil. Instead of writing diamine, you will write uracil. So you will not have what? You will not have diamine, and therefore the diamine position is replaced by uracil, and therefore this one diamine links with adenine, and guanine links with what? Cytosine. Cytosine links with guanine, and adenine will link with diamine, but because it is an RNA, we are forming the RNA, we are supposed to put a U. There is a uracil, and this one will have another uracil because a diamine will always be replaced by uracil, and therefore this is the what? The RNA, the RNA formed. The RNA formed. The RNA has been formed, and you must be very very keen to understand that this process takes place in the nucleus, and therefore we can say that in the nucleus there are some RNA, and also there is DNA. So RNA and DNA are found in the nucleus because the process takes place in the nucleus. And after the formation of the RNA will get out of the nucleus into the cytoplasm. And once it gets out from the nucleus and enters into the cytoplasm, it is referred to as mRNA, that is a messenger. And therefore, this one RNA, it will just come out, it will get out, and therefore it becomes very free to move within the cytoplasm, so that it can go to where here on this side we have adenine, we have uracil, and here we have adenine and cytosine. You can see it is well illustrated here. Then we have guanine, then we have uracil, and finally you have uracil. And therefore you can see in this diagram, we have the resulting RNA, and this is what we refer to as mRNA. Why is it called mRNA? Because it acts like a messenger, and therefore it will act like a messenger, whereby it will take the information from the DNA, that is from the nucleus in the DNA, and it will go to where to the ribosome, where the process of protein synthesis takes place. And after that, there is another RNA which is involved, and the second type of RNA is tRNA. And tRNA, it is also an RNA, it is a type of RNA, and this helps in translating the information from the messenger RNA, so that it can be taken to the amino acid pool, so that the correct amino acid can be removed from the amino acid pool for the assembling of the protein which need to be synthesized. And therefore, this RNA, we have it translates the information, the coded information from the RNA, that is messenger RNA, into so that the assembling of the correct amino acid can be assembled from the amino acid pool for the formation of the protein which is needed to be manufactured. Having said that, I'm going to give you to explain to you that it is very, very important to know that the nitrogenous base occur in triplets and in a pair of three, and the pair of three is what we refer to as a codon. and the codon is coded. The codon is normally coded for the synthesis of a particular amino acid. So we can come to the type of amino acid. So we have several codons, and the several codons which they are there. And before I give the different examples of codons, I will also wish to highlight that there are two types of amino acid, and the amino acids are the building blocks for protein. And therefore, in order for a particular protein to be synthesized, there must be several amino acids being linked together so that we can form a complex molecule chain called protein now. And therefore, as far as the codons are concerned, we have two types of amino acid. We have essential and non-essential amino acids. Essential amino acid are the amino acids which the body cannot manufacture, and therefore it is very, very important for you to supply them to your body through dieting. So that you can provide them into your body, or you can introduce them to your body through what you eat. And the non-amino acids, it is a category of amino acid which you don't need to eat because the body is in a position of making its own. And therefore those amino acids whose body is capable of manufacturing are what we refer to as non-essential amino acid. While essential are those amino acids which the body cannot manufacture, and therefore you have to introduce them into your body because the body is unable to manufacture them. So as far as the codon is concerned, I have a codon represented by letter A, three A's, and therefore as far as three A's are concerned, it is coded for the amino acid by the name phenylalanine. Then we have the second one is TTT. It is coded for the assembling of the amino acid lysine. And the third one, we have it as CAA. It is coded for the information for the assembling of amino acid. These are essential amino acids, that is valine. So the first one, that is phenylalanine, and lysine and valine are essential amino acids. And lastly, we have CTA. It is coded with the information for assembling of amino acid by the name aspartic acid. And therefore, these amino acids are very, very important. And once they are assembled, they all help in synthesis of protein. And remember, protein is very, very important into our body because it forms the basis of the interpretation of the characteristic of living organism. And therefore, as far as the three codons, the four codons are concerned, I'm not saying that they are only four, but they are different types. There are different types of codon, but in this lesson we will only discuss about the four types. That is the four types of codons, and that is AAA, then TTT, CAA, and also CTA, and also the respective amino acid which they normally assemble from the amino acid pool. And therefore these amino acids are very, very important because they are involved in the synthesis of protein, of protein. And now I'm going to come to the next area, and I'm going to explain the differences between DNA and RNA. So the next area that I'm going to touch I'm going to touch on differences between DNA and RNA. Differences between DNA, and RNA. So DNA and RNA. So I'm going to, on one side, I'm going to write DNA, and on the other side, I will write the RNA. So DNA, the first difference I can write it is DNA are found in the nucleus. Found in the nucleus, that is the DNA. And as far as the second one is concerned, that is as far as the second one is concerned, that is the RNA, found in the they are found both in the nucleus. Found, that is number one, found both in nucleus and cytoplasm, in the cytoplasm. Then the second one, the second difference is that has deoxyribose sugar, has deoxyribose sugar. And the other one, number two, has ribose sugar. Has ribose sugar. And the third difference is it is a double helix, exists in form of a double helix. That is there is Li, double helix. And number three, as far as the other one is concerned, it is single-stranded. Single-stranded. So it is supposed to be single-stranded, and therefore you can look at them and you can see that those are the differences that we have between the DNA and the RNA. And having said that, I have come to the end of today's lesson. Your presenter was Mr. Wanyoike, and you can reach me if you want to, you have any queries. You can reach me using the following numbers. The cell phone number, that is positive 254, that is the Kenyan code 254901639. So you can use this number, that is positive 254. Sorry, there is I've omitted 3 numbers: 724901639. So you can use positive 254, that is the Kenyan code, the country's code, that is Kenya. The code for Kenya is 2, positive 254. Then you followed by my number now, 724901639. So you can use this number to reach me for any question. You can always contact me using this number. Also, you can search me in the YouTube using the name Mr. Wanyoike, and once you you search me by the name Mr. Wanyoike, you will find me there. For more info, till next lesson, I'm looking forward to have a very wonderful lesson and next lesson, and I hope you are going to digest the already tackled question. And please subscribe to my YouTube so that you can always be informed each and every day. Thank you.